Thank you, Joe. Thank you all for your attention and sticking around. So my name's Oliver. I'm so happy to be back in Korea. I used to live here for a little while. So yes, it's a great pleasure to be here. The cellscape is the baby of my laboratory. And as Joe said, it's part of this spatial ecosystem that we have. And when you're doing the research, the type of discoveries that Joe talked about now this afternoon, looking at whole transcriptome or 570 proteins, you make discoveries. Sometimes you make discoveries on accident, but you're almost guaranteed to find something new. But there comes a time when you need to translate these findings into potentially a diagnostic assay, or you need to convince your pathology or clinical colleagues down the hallway that what you found now is important and you need to translate that. It turns out that multiplex immunofluorescence or histology, as it has been done for a long time, is still the one means that we use to diagnose diseases. So you need a tool, you need a vehicle to take all the findings out of these two boxes and bring them closer to the clinic. And that's what we think is doable with the cellscape. So I'll show you what that means and how we do this research and how the system works. And really, I think the tagline here is from discovery to translational research. I spend a lot of time in my life doing discovery research, but my focus is really how to make findings, quantifiable. How can you measure things in large cohorts, for example? So let's go back to Miranda Orr's study. When we first met Miranda Orr last year in October, this is what we had. We had a volcano plot with hundreds of proteins that were either downregulated or upregulated. Lots of stuff going on here. Not every one of these proteins will ever be studied. Many of them don't matter because they're below the range. But using a volcano plot, whatever's up here that's upregulated, or whatever's up here that's downregulated, those are the handful of proteins that people remember. Those are the handful of proteins that appear in their conclusions in their papers. Those are the subjects of PhD theses. Those are the targets, biomolecules that may someday become druggable. Now, how do you solidify this type of a finding? Well, as Joe said, in Miranda's case, we found about 30 biomarkers in these types of data that we thought could be very important in certain neurodegenerative diseases, including Alzheimer's and ALS. The question now became, outside of a one-off or two-off study in which we found many biomarkers, how frequent are these proteins expressed? What cell types are they expressed? And very importantly, if you take a large brain section like this one, where across the brain are they expressed? A brain is not a homogeneous piece of tissue. It's a highly regionalized, highly diverse piece of tissue. So we need a system that's agile. We need a system that allows you to do high or reasonably high throughput. And we needed a system that allowed you to generate data on a large canvas where every single pixel on that canvas, you can zoom in and out, do whatever you want, but you can quantify your data. That's what cellscape does. So in the first experiment that I'll show you, the experiment that we did with Miranda, it was our job to build a 30-plex panel with 20 unknown targets, 8 of them being phosphorylation targets in proteins, which is really hard to label in less than a month. When I first heard about that, I thought my boss is crazy, and I said, how are you going to do that? But it turns out we did it, and we did it easily, and we've been doing this day in, day out since we developed assay chemistry before that. So here's what these data look like. This is what we call quantitative spatial phenotyping of phosphorylated tau targets in AD, Alzheimer's disease, and ALS. On the left here, you can see tissue from an Alzheimer's patient. On the right, you can see tissue from an ALS patient. We're just focusing here on the dentate gyrus, really just a fraction of this massive piece of tissue that we looked at. We're now looking through different types of targets. Here's neurogranite. You can see beautiful mosaic expression of this marker in certain cell types. High expression, low expression, yes or no, there's all kinds of information we collected here. Interestingly, this marker is expressed in AD, but not in ALS. I can go through here now and narrate all kinds of interesting findings that we did. Another one, for example, phosphorylated tau at serine 214. It's actually not in the dentate gyrus, something we thought it was. Turns out that it's in glial cells in ALS. These are unexpected findings. We're not yet translating them, but you can see just the richness and the disparity of data here. So the cellscape is a great vehicle to generate single cell, quantifiable, extremely high quality data from any type of biomarker profile that you previously done. Now, a couple more features about these data in this experiment is the resolution of the cellscape is 180 nanometers per pixel. That means you can easily photograph a cell. But you can also photograph the geography inside of this, essentially the subcellular buildup. Joe talked a little bit about neurofibrillary tangles. Well, this is a neurofibrillary tangle right here in pyramidal neuron. Here's another one, here's another one. Here's a vesicular accumulation of this tau protein, and here's what appears to be a paracellular localization of ubiquitin molecules. What exactly this is and what it means remains a subject of investigation. But these are the types of findings that one starts to look at when they're building spatial signatures and trying to understand more and really have data points that track and coordinate. So this is spatial biology. This is not so much about the complexity of an experiment, how many biomarkers you can see. This is neuromorphology. What this bottom images show here, I think is also very, very important to remember about histology and immunofluorescence in particular. In this brain, you have neurons in yellow. The sick neurons here have these really massive tangles. This neuron's probably dead. This is probably dead, dying, gone, just starting to. So you can do all kinds of interesting analysis. But what's important always, and you cannot really do this reliably with any type of transcriptomic technique, is that the extracellular compartments are generally as important as the cells, especially in the brain. This here's an amyloid plaque. It's a hallmark of Alzheimer's disease. Recognized 100 years ago. To this day, we diagnose Alzheimer's disease in deceased patients based on the presence of plaques. No transcriptomic technique, especially not in this detail. But with the cellscape, with the right collection of biomarkers or antibodies, you get to stain all that. You get to map all that. It doesn't matter if it's a dying or dead neuron, one that's just recently unhealthy, or if it's extracellular neurofibrillary tangles or amyloid plaques. All of these data are captured in our images at 182 nanometers per pixel. So as Joe said earlier, I think there was some result that you could talk about for 3 years with the whole transcriptome. I could probably talk for 3 years of just about tiny little cellular granular details out there. And yeah, this might be a translational vehicle, but there's still some discoveries that can be made. So this gives you a bit of a flavor of the type of data that we produce, what we can do. It's really a comprehensive capture of the protein landscape, spatial proteome of your tissue sections. How does this experiment work? It's simple by design. We wanted to keep this simple because, as I said earlier, it needs to be agile, it needs to be modular. It should be easy for the end user to adopt. So let me walk you through this workflow. This here is a cellscape. It's about the size of a relatively large desktop printer, but it's a benchtop system. You don't need any special infrastructure for this. It's a microscope in there, and I'll talk about that a little bit later. Let's focus on the assay first. This system is designed to be compatible with standard histology slides, which you can see here. You tissue on a slide, you put a cover slip on top of that, that's this blue thing here, and then seal on with an adhesive. This cover slip has a hole for inflow and a hole for outflow, which allows you to deliver fluids, reagents, buffers, and hydrate your tissue. Once you have done that, which by the way you do after dewaxing and antigen retrieval, your tissue is sealed onto the outside and that cover slip is hydrated. And it's very stable for many, many months. I'll show you a data point for that in a second. Now here's what I think is one of the very neat parts about this cellscape. In some sense, this is an auto-stainer as much as it is a microscope. So why am I saying that? It is an auto-stainer because we put the tissue on without any antibodies on it yet. The antibodies are delivered by the system in the beginning of the cycle. In our case, we like to use fluorophore-conjugated antibodies. So you're going from an unlabeled to a labeled tissue. Now you're taking a photograph, capturing, say, 4 biomarkers per cycle. That's generally what we do. And then we use photobleaching to remove that signal. So what this means is that your antibodies, your first 4 antibodies that you delivered, they stay on here, but the fluorophore signals are gone. I'm going to speak about this in a little more detail because it's unique to our system and how we do it. There is no other system that deploys photobleaching the way that we do. so it's important to understand the nuance. But before that, let me just talk a little bit more about this process, because I think this is something that excites us. We talk about this a lot, and I think it's important for you to remember that when you're in translational biology, you need to be agile, you need to be modular. You need to be able to make a series of left, right, yes, no decisions. Is my tumor hot? Yes. Oh, if it's hot, then I might put this antibody instead of that antibody. We see this all the time. Spatial biology is not always predetermined fixed panels. People change their mind every day, every hour. So this is what our system allows you to do. I've just walked you through how that works. So let's say we stain our tissue with four markers, 10 markers, however many markers you want. You can take a stop, you can take a pause, halt, look at your data, analyze it, discuss it with your colleagues. And meanwhile, you're taking your sample and putting it in a fridge. It just sits in the fridge, and it's been on for weeks and months. And if there's something about this tissue that is worth further exploration, for example, very simple problem, the quality is good enough. I would encourage you to always do this. Always test your tissue first with 1 or 2 antibodies. Don't commit 100 antibodies to a tissue unless you know if it's good enough. There's a lot of bad tissue out there, believe me. But say you want to do more work on it. You take that same sample out of the fridge and put it back on the microscope and start restaining it, start reimaging it. It's up to you. You can do that, do it over and over again. And what does that look like? Oops, wrong button, sorry. Here's one example of what that looks like. Similar to an experiment that Joe just talked about with fibronectin. Turns out that the extracellular space is also very important in regulating what the immune system can or can't do. Here we have a ductal breast cancer, a grade 2 invasive carcinoma. And we stained this with, I don't remember, it was about 15 antibodies at the time. We were just doing basic immune profiling. Again, this is multiplex immunofluorescence data, spatial proteomic data. You can see the tumor mass here, blue and white, and outside of it, there's this rainbow of immune cells. Now, we saw this picture, and it's certainly beautiful, and we can see many, many immune cells that we can catalog. But the question was, similar to the question that Joe asked this morning, what is this gap? Why is there a gap? I think Joe said Mother Nature doesn't like gaps. So a few weeks later, we took that same sample out of the fridge again, and we stained it a little more. We put some architecture markers on. Now look at this. There's no fancy image registration required. I could generate these data. I'm a bioinformatician. I'm not bioinformatically inclined. But this is a perfect continuation, a perfect completion of tissue biology in plain sight. There's a layer of plasma and epithelial cells around here that is most likely preventing the migration of the immune cells in the day. This sample is still in my refrigerator. I can now probably keep poking around on it if I want to, but I just want to explain this feature to you because this is absolutely unique to these spatial biology platforms. If you want this to be, it can be a stop and go system. It's up to you. Let the tissue guide your research. And it's extremely important, especially if you're doing this type of translational biology, where really time, money, and those types of things are of the sense. So I think at this point it's a good time to talk about photobleaching, because you need an assay that enables you to do this work. Let's say we were working with a barcoding strategy, or so generally that requires that we put all of our antibodies down at once, with some assumptions we need to make. But the reason we use photobleaching is it's easy to use in principle, but it also allows us to design these types of modular stop and go assays. Now, for those of you who know about photobleaching, you may agree with me that this might be the most pragmatic, but somewhat limited way to remove a signal. Remember, in our experiment, we stain a tissue, stain with 4 colors, then we need to remove the sample. We need to remove the fluorophores, and then we either park it in the fridge or we keep going to make a high-plex assay. It's really a choice. Up until recently, these types of techniques have been quite limited by the type of fluorophores that they can use. BUVs, FITC, PE, or CP. These are not very bright, they have bad signal to noise, but they photobleach easily. So it worked, but it wasn't good enough. We always wanted to work with modern fluorophores like Alexa Fluor 594. Alexa Fluor 594 is probably the most stable and hard to remove organic dye that people use in histology. When you shine a light, in this case a white light from 400 to about 700 nanometers on Alexa Fluor 594, can see that the dye starts to bleach, but it never really bleaches fully. So it takes a long time, it never really goes away. This means that up until recently, people who wanted to do these experiments could not use these types of dyes, which is an absolute shame because this dye is extremely bright. It allows you to see really hard to detect targets. Now, I'm happy to report that my lab has a guardian angel or something that gives us luck every now and then. I think I've been quite lucky in my lab a few times. And we made a discovery a while ago that is now called the EPIC-IF technology, which I think we had a fair share of luck, but I think the timing and really the execution on this has been awesome. EPIC-IF is a way that allows us to enhance photo bleaching. So the problem that I've just described to you now has been solved. I can take any organic fluorophore that's out there, you know, Alexa dye, dialite, biotypic, coralite, you name it, whatever's available in the major antibody analogs is going to react to this. This is a buffer that we add to our photo bleaching step and then effectively remove any one of these dyes in a very short amount of time. Now, I'm going to show you some videos and show you the behavior of these dyes when you photo bleach them with or without EPIC. You can see here, the curve already gives it away. This is trying to bleach Alexa fluorophore 88, labeling CD3, an enormously strong signal. Without EPIC, doesn't really help. It's not really useful for multiplex experiments. And here's what EPIC. Now this is what the video looks like. See, beautiful bleaching. The only thing that's left are RBCs, erythrocytes, and other tissue autofluorescence, which this system is not designed to remove. That is something we remove with background subtraction later. This here is my favorite example. This is Cytox Orange, nuclear dye. You can see every single nucleus is labeled in this tonsil. Super bright, ubiquitous. Watch closely. This is super fast. So now you can think about building an accent. It's up to you what you want to do. You can stop and go, you can bleach something quickly, you can bleach. One of the nice things about this that I don't have a slide about right now is this system will selectively remove a dye based on its color. So let's say you have a tissue labeled with red, green, blue. You can turn on the excitation light, but only in the red channel, remove the red signal and let it stay. So it's really, in terms of spatial platforms out there, this is probably the one that allows you to be most creative. Actually do experiments that you may need to design, you may need to customize. I think that's something we are not overlooking on this one, and that's what makes this system unique. A very important precondition that we need to fulfill, assumptions that we cannot make without testing them, obviously, is that does this system affect the tissue or reagent quality? It does not. We spend a lot of time in my lab testing this, so we do a lot of different photo bleaching experiments. No photo bleaching, without EPIC, with, and very restricting. We routinely H&E stain this stuff and get it scored by pathologists. Today, we have never received a report that would indicate that the system is damaging to our tissues. Most recently in my lab, we have gone as far as 55 cycles with EPIC-IF. So 55 times 4 antibodies is 220 markers on a tissue. And we are, in the last cycle, still able to detect weak targets at PD-L1 plus T3. So I can tell you, I can report to you today, and you're easily able to go past 225 markers with the system. This is an immunofluorescence. Every single marker here is going to be labeling what it should be doing after 225. I don't know what the maximum is, but 225 is more than most of the people do that I work with. Now, to summarize, there's one more last point that I think I need to talk about. And with EPIC, with photo bleaching, you have a lot of freedom to move when you're building a panel now. Just think of it. The three big antibody vendors in the world, let's say Abcam, Thermo, and maybe Protein Tech. We did some research, and we looked to see how many antibodies they had that were already validated for immunofluorescence that carried a fluorophore already that was compatible with our EPIC system. It turns out there are more than 6,000 antibodies readily available for that particular purpose. So if you're looking for an antibody today, before you even ask me, you can go to the Abcam catalog or the CSG catalog, see if it's conjugated 488, 555, or whatever it is, and if it is, and it's IF validated, there's a more than 85% chance that it will work out of the box from a third party, Abcam or someone, on our system. So you do not always need to obtain the antibodies from us. That's a huge benefit. It allows you to move faster, and it allows you to choose targets that you want. We do not prescribe a target to you. I think that's one more very, very unique thing about this. So enough about the assay for now. Let's talk a little bit about what this microscope does, what makes it unique. Let's talk about the actual cellscape. This is what it looks like. Again, this is a benchtop system. This is our microscope. So in here, you have an inverted epifluorescence microscope. The lens comes up like that. The light path goes here and goes to the back. On the stage, fully automated, a Mortzhauser stage, moves very precisely. We have 4 slides. 4 slides. You can see here, the cover slips are already on. There's a TMA, there's a tonsil and another tissue. Connected via a microfluidic system to that fluidic device. This is where all of the staining reagents, antibodies, buffers go to. So it all comes as one. The data that the cellscape generates are fully stitched, shading corrected, and curated. At the end of the day, OMI-TIFF comes out of it. OMI-TIFF is a very highly compatible format, loaded in 2 paths, gives you farm payload. There are many different ways that you can analyze, look at or analyze these data. It was designed to be plug and play. That's a strong statement to make. Nothing's ever plug and play in spatial biology. Let's go over this system. There are 3 features that we want this microscope to have that we think it differentiates. One is, and I already mentioned it, the resolution on this system is 180 nanometers per pixel. It's extremely high for an epifluorescence microscope. What I mean is these data are acquired with a 20x lens, so you actually get a really large surface area with a really good resolution. I should say this is not optical resolution, it's digital and resolution, but the data that you get out of it are absolutely stunning. We generate all of our data in high dynamic range, something we call HDR imaging. I will explain to you how that works in a second. And all of the data are captured on this slide, in this blue area here that you see. So this is a cartoon of our slide. You see how it works. Here's the inflow. Reagents go through, come out. And anything that's in this blue area is real estate that you can use any which way you want. In this case, there's a multi-tissue array. There's a tonsil, there's an appendix, ovary, I think, thymus. This is the type of tissue that we use in the lab to test things. We can put a TMA with probably several hundred cores on here, or a massive tonsil. I would estimate that if you took a dense tissue like a tonsil, thymus, or something, or B-cell carcinoma, you could probably put up to 7 million cells on a single slide. That's a lot of cells. That's more data than most single cell techniques to this day still generate, except for the ones that you'll reach designs. But yeah, you can really, really generate a lot of data. Again, at 182 nanometer pixel resolution, that's a lot. Here's what it looks like when you have 182 nanometer pixel resolution. This here's a cellscape image. This is a T cell expressing FOXP3. A couple things to notice. FOXP3 is no longer just a pink signal inside. You actually have compartmentalization of that signal. What it means, I don't know. It's potentially interesting. But look at the sharpness and the crispness of these plasma membranes and compare it to what it looks like when you take a picture on a competing instrument. These are okay T cells, it's fine. You can circle them, you can measure them, whatever. But the amount of bleed through into regions that's not the plasma membranes is probably unacceptable to a lot of bioinformatic algorithms today. Your system or your bioinformatician or whatever programs they are using to count these cells is going to be much, much more prone to error on this type of data than on that type of data. And going back to saying, hey, we capture 7 million cells on one slide, say you have 20% error rate or something here, you don't have that problem. Even a 5% error rate at 1 million cells can really skew your data one way or another. So resolution is not a trivial problem. It's important to have extremely high optical resolution. The other thing that contributes to the crispness and sharpness of our images is this HDR imaging. So HDR imaging is essentially a series of exposures that our microscope captures at every scene as it goes over the tissue. Our microscope doesn't just take one image, it takes several. And usually it starts with a high exposure, trying to capture all of the expression of all the molecules, and then works its way downwards in an algorithmic manner that I cannot disclose fully until there's nothing left to capture. The outcome of this is that high and underexposed regions can ultimately be combined into one high dynamic range image. We have extended the range of our digital sampling device. It allows you to take a cell like this T cell, or this mobile site and really try to capture the full gamut of biomarker expression within it. In this particular instance, you're overexposing it. You will not be able to measure much here. Bioinformatician is going to reject that one. Here, looks pretty good. Here it's too dim, and here it's too much overexposure. But in your HDR, you find that this problem has been solved. So it extends your dynamic range, but it also allows you to handle a workflow more easily. We just got an email today from a collaborator who spends a lot of time imaging high throughput, high quantity stuff on slide scanners, some of us in the market. And he started to work on this system, and his response like, wow, I can actually take a picture without optimizing anything. That has never happened to me before. He was saying that it takes him months and months and months to train his postdoc to take a good picture. I'm sure we're all mindful of that, especially the microscopists in the room. With the system, you don't even need to do that because it takes that problem out of your hand. So not only does it increase your data quality, but it streamlines your acquisition. It makes your life a lot easier. That being said, our system generates a lot of data. In comparison to other systems that just take one exposure, we produce 4 or 5 times more data on average. Historically, that's been a problem, but we've solved that with the introduction of power omics, which is a new data engine that lives inside of our microscope. So this is a bad thing. You would not be exposed to that. What we see here is PD-L1 staining in a standard single capture experiment. This is what you would get from standard slide scanner or our competing instrument, for example, versus the cell-scale power omics. The signal to noise in this particular image, I believe, is about 8 times larger in this one over that one, something like that. On average, I think here, we measured the signal to noise and compared them for about 35 different biomarkers. On average, our system produces a 2.5 higher signal to noise rate than a competing system. If you're working with a hard to quantify biomarker like PD-L1 or FOXP3, this is very important. You have a 2.5 better chance of capturing this biomarker. And anyone who's ever tried to find PD-L1 expression in a low expressor would probably be excited to hear that because that gives you a better shot at getting the right data. All around or taken together, even though we generate so much data, we've managed to curate them and to process them in a way that the size of these data they have parity. They're equivalent to what you would see in a competing instrument. And the acquisition of these data, the digital processing, has also approached parity. So you get better data, but you invest the same amount of time and money to generate. I think it's extremely important, and it's also very exciting. So let's get back to assays, and I'm going to talk about 2 applications, 2 experiments that people have done on this system before I wrap up. We make kits. They are kits that are readily available for our end users. We don't force you to make them, but I think they're designed in a way that they're small enough that I think anyone who gets a cellscape who works in cancer biology or any immune-related problems, and soon also neuroscience, will find use in them. So what you can see here, so these are in inventory, they're ready to go as of today. We have kits for segmentation, super useful because it stains the membrane and the nucleus. So if someone is using an advanced segmentation algorithm in a lab, I highly recommend that because you get excellent segmentation data from this. We have a spatial immune profiling kit designed for immunologists and cancer biologists. It allows you to detect all the cell lineages plus a few checkpoints and activation states. In addition, something that I think immunofluorescence absolutely shines with is the tissue architecture. Again, most of these things you can't really detect reliably with RNA research. So these come as they are here, and importantly, they're plug and play. So if all you're ever interested in is tissue architecture in your research, you just need to get this, and you can work with it. You don't need anything else. Or if all you care about is immune profiling, you do that. But if you want to do all 3 together, you can do that as well. And you can slide custom analysis. I'll show you an example of that in a second. So these are the kits I've just talked about. And in addition, we're working on a myeloid kit right now, actually. It should be out at some point. But I think the philosophy that I've developed over the years that I've been working with this stuff is I can never build kits as fast as I need to make everybody in this room happy. Because everyone's going to have their own antibody that they need. So this is where the customization and the drop-in becomes important. And I'm going to go back exactly to what I said earlier. There are thousands of antibodies available today that have been IF validated that you can use on the cellscape without having to talk to them. They are available for human FFPE tissues, they're available for mouse, fresh frozen, FFPE, you name it. Whatever the antibody companies have, is High-Fidelity has an organic fluorophore on it, trying on the cellscape is extremely high likelihood that it will work. This allows you to start an experiment quickly on a kit, like immune profiling, that's the thing, but you find that I don't have good NK cell markers or not enough myeloid cell markers, you go buy your own and put them into that assay because it's that easy and it allows you to move fast. That's what we want to do. Customization, agility, mobility is key. That's what we really hope that we can achieve with this system. And the last two things are two examples in which exactly that was done. People have used the cellscape, we built an assay that we have not preconceived, that we don't have to inventory. We've changed the rules a little bit, and they learned something new. The first one of these experiments is Jasmine Plummer. She's at St. Jude's Pediatric Hospital in Memphis, Tennessee. And the problem here is, in St. Jude's, they have an interest in treating solid pediatric tumors with CAR T cells. The story is actually a sad one because generally these kids that have these tumors that get CAR T cell injections, that's a last resort. And they don't do that up front. This is really a whole clinical trial. But one of the issues that she has is, or that St. Jude's has, they don't know what happens to these CAR T cells once they're delivered. Where do they go? Do they actually go to the tumor? Once they get to the tumor, do they infiltrate? Once there, what do they become? CD4 T cells, CD8 T cells, we don't know. The other issue that, so Jasmine approached us and said, can you help us? Can we start to visualize these T cells in our tumor model? And I said, sure we can, but the problem is we didn't have a CAR T marker. And it takes a long time and costs a lot of money to raise an antibody for CAR T. So what Jasmine did, we did together in collaboration, we found a company that would raise a fish probe for us using a single molecule fish against the CAR sequence that's specific to St. Jude. It took about a week to make that probe. It's a company called Molecular Instruments. Now, all we needed to figure out is how do we incorporate a fish assay onto the cellscape with our immunofluorescence, same section. Well, it turns out it was very easy. What we did is we stained the tissue for the fish probe first, left the fish probes down, used EPIC-IF to remove that signal, and then stained it with antibodies. And this is what you see here. This here is just the antibody staining of the mouse xenograft sitting under the skin right here. There's the tumor in white. You can see there's a margin, and this is sort of the outside area. This tumor was implanted, I think, 3 weeks before we cut it out. So you can see the immune system of this mouse is reacting like crazy. There's a ton of CD8 T cells here, CD4 cells, and lots of enzyme B inside the tumor. Nice. It means that the system's working with an immunological response. Now, getting to the CAR T, you can see here, this is an RNA fish probe on the same section done on the cellscape. Remember, the same section multiomics. Everything that's red up here is a CAR T cell. We have now answered 2 questions already. Does the CAR T cell in this model reach the tumor? And the answer is yes. Does it infiltrate the tumor? Yes, it does. Not to the degree that it's sitting outside of it for reasons we don't know yet. Probably another extracellular molecule, but it goes into the tumor. Once it goes into the tumor, what does it become? Here's where the resolution of the single-cell biology of the cellscape comes in clutch. This here in green is a CD4 T cell. Very, very clearly, it's a CAR T cell. It's a beautiful expression of this fish probe. Turns out that this cell is also labeling for enzyme B. So the CAR T cell, in some cases, not all, turns out to be an immune regulator, an immune stimulator. So when CD4 T cells have enzyme B in it, they tend to stimulate probably killer T cells in your surround. So it's very possible that this cell is sending signals out to all these yellow T cells and say, go attack, go kill, do this and that. So we now know what this CAR T cell line is doing in this particular mouse model. They had zero idea of doing that before. And all it took was run a couple of assays, same fiction multiomic ones on the cell scale. So last example, I think I'm going to use this to wrap up, just to get back to this idea of customization, modular panels. I think this is really important. Once you have a hypothesis, once you have an idea, once you want to study something specific, you need a platform that allows you to do that. That's exactly what Nick West has needed. Now he has it. So Nick's been studying these nasal polyps for some time. So he's an immunologist. And the idea here is that when you have chronic inflammation of the nasal passages of the airways, You start to grow these little things, your nose is itching, whatever, but architecture of your nasal passages, your airways is remodeled. You cannot control that. The immune system is causing your nose to do things that you probably don't want. Nick has very specifically focused in on eosinophils and M2 macrophages. Those are two cell types he wants to study. Now, if you look at my panels that I've developed, there are few macrophage markers on and absolutely no eosinophil markers. So we had to customize, we had to add more antibodies to help Nick to get to the answer that he wanted. And this is what this is. Now I'm going to show you, I'm basically just going to walk you through a time frame on this experiment. This is an experiment that's still in progress. Nick and I are like back and forth. He lives in Australia, I live in the United States. Days go by before we come to any consensus, many cups of coffee are consumed. But I think this experiment to me really outlines the beauty of the experiment. Anytime I work with a new person, he was new to me a few months ago, I say, look, I don't care about your tissue, it's good or bad, but I want to see what it looks like with my antibodies first. Let's do some basic immune profiling. And that's what this is. This is basic immune profiling of a nasal polyp. See blue B cells, CD56, you see lots of NK cells actually in this tissue, it's quite interesting, and CD4 T cells. Seems to be that there might even be these tertiary lymphoid structures of these polyps. Super interesting. I thought this was a great experiment to do because it confirmed to us that this tissue has good quality. It confirmed to us that it's compatible with our assay. It's a basic assumption everyone always has, a basic question I always get. Well, Nick wasn't yet quite animated because, of course, he's interested in eosinophils and so on. You can also see this already lent itself to some analyses. But if you think about a nasal polyp, it's as much an immune problem as it is an architectural issue. So it made sense, of course, for us to follow this experiment up. I should add several weeks later with some architectural markers. So here you can see an epithelial marker, beta-catenin, meat-heron. See it really nicely all over the place. The vasculature, this thing is actually highly vascularized, which means it's quite old, very established. So the only way to remove these things at this point is probably surgery. Again, you don't want this thing growing in your nose. We know that for sure, but this is a very advanced one. But what is striking about this is the infiltration of epithelial B cells. Again, B cells. We never expect them to do all this. It's supposed to be educators in lymph nodes or something. But it turns out they're actually quite active when they're unleashed in peripheral organs. So this is quite interesting as it's a new finding. But this was an experiment that was done weeks after the original experiment. We went in and we started to analyze it a little bit more. You can see a picture develop now. These are cellular neighborhoods. Essentially, these are unbiased ways of looking at groups of cells that are talking to one another. It's not just simple cell counts, it's coincidence detection of different types of immune cells. But now, last but not least, and most important to Nick, is this idea, okay, let's stain this tissue with eosinophil markers and macrophage markers, M2s, M1s, and other types of myeloid cells. You can see absolutely gorgeous staining of these eosinophils all over the place. These yellow neighborhoods, I think, are primarily the ones that contain them, and 5, so labeled with CD66B and CD15. I did not have these antibodies developed in my lab. We bought them from a third-party vendor. This is what Nick wanted to study, and now he has it here. Beautiful single-cell resolution, massive pieces of tissue. We're imaging a whole cohort right now and analyzing it. Hopefully this will help us to generate a better understanding of what's actually going on in these organs and how this problem arises originally. But that to me really captures how experiments can be done in stationary. It's an organic process. The tissue is guiding us. The tissue is challenging us to ask additional questions, to take next steps. We're never flying blind here. We can always keep an eye on the tissue and we can make decisions. Do we go? Do we go on? Do we stop? Do we go back to right? That's really, really nice thing about the system. And I think that captures what we would like to do with it, or we encourage you to think about as you think about these types of experiments. So, 감사합니다.